All right, so this is just a quick little few-minute presentation on something that we didn't quite get to the other day, which is how does the body or how does the immune system recognize self from non-self? And so the first thing that you need to understand is that all cells throughout the body are constantly presenting internal proteins to the outside world, to the immune system. And so on the surface of every cell, there's proteins that are specifically designed to display the proteins that are inside. You can think of this as like a showcase in the front of a store like Zara or any of those other department stores. You can see what's inside. And, um, you know, there's an ability then for the immune system, if there's something that's recognized inside that isn't supposed to be there, then it destroys that cell. And so what we're going to do right now is we're going to take a look at the difference between helper T cells and cytotoxic T cells to understand why cytotoxic T cells destroy the cells when they find something wrong, and why helper T cells become activated when they find something wrong inside of a cell. Alright, so let's start off with your helper T cells. Remember that their job is to basically sound the alarm for the immune system. So a helper T cell often gets a signal that something's wrong from a macrophage. But if you have a helper T cell here interacting with a macrophage, and the macrophage is presenting an abnormal protein to the helper T cell, you don't want the helper T cell to kill the immune system cell. Okay, that would defeat the whole purpose of the generalized immune system. If the helper T cell was killing this macrophage, now there's nothing to do the phagocytosis, the heavy lifting of the immune system. So instead, what happens is there's a system in place where the helper T cell is able to read what's inside the macrophage, but doesn't destroy the macrophage. Rather, it's activated by the presentation of these abnormal proteins. So what we're looking at here is the binding of the two cells, and the binding of a helper T cell with a macrophage or phagocytotic cell of the immune system. It's all done through these proteins on the outside called MHC2 molecules. And so an MHC2 molecule is just a protein, and these two proteins, one from the macrophage and one from the helper T cell, can interact with each other. Now what's happening in the middle of this interaction is that the macrophage is showing the proteins that are contained within inside of it to the helper T cell. Now if the helper T cell were to recognize this antigen as being something foreign to the body, it would then become activated and go through clonal selection. After going through clonal selection, some would become helper T cells that would be effector cells, and some would become memory cells in case of later infection. Now there's a special chemical called interleukin, and what interleukin does is it's a chemical secreted by the macrophage that tells the helper T cell to get close. Okay, so it's drawing it in, and when an activated helper T cell is present, it will also release interleukins to attract the attention of cytotoxic T cells and cytotoxic B cells in the area. Basically, it's sounding the alarm, saying something's wrong. This cytotoxic T cell comes close. Is it something I recognize? If it doesn't recognize, it goes on its way. B cells come by, check in on the helper T cell. Is it something I recognize? If it is, then this B cell will become active and then go through clonal selection. Now, one of the other things that's different between the interactions between a helper T cell and a macrophage and a T cell and a regular body cell is that helper T cells have this protein called a CD4 protein, which is helping facilitate this interaction here that happens between the MHC2 molecules. So let's see what happens in a cytotoxic T cell. Alright, so here's a diagram where you see cytotoxic T cell in action. So remember, killer T cells or cytotoxic T cells are not responsible for sounding the alarm of the immune system. They're going out and they're destroying cells that have something wrong with them. So, as we said before, all cells of the human body are able to present the proteins within inside of them to the cell surface. This, in most cells throughout the body, is done through MHC1 molecules, similar to the MHC2 molecule but a little bit different. So the MHC1 molecule of my regular body cell interacts with the MHC1 molecule of my killer T cell, and if the antigen inside this infected cell is presented to my cytotoxic T cell, and if it stimulates or activates a cytotoxic T cell, then we're going to have the cytotoxic T cell release this chemical called perforin, which punches holes into the cell membrane of the infected cell, causing it to lyse and preventing it from building any more viruses that could infect potentially other cells. Now, one of the other differences that you see here between a cytotoxic T cell and a helper T cell is that the MHC1 molecules of these two cells is being held in place by a CD8 protein. So similar in function to what a helper T cell has, a little bit different in terms of structure. So this is what makes a helper T cell, the alarm of the immune system, and the cytotoxic T cells, the destroyer of the immune system. Helper T cells trying to stimulate other cells of the immune system. Cytotoxic T cell is doing the actual job. It's doing the dirty work. It's getting rid of those cells that are affected. And so the way that they're able to do these different things is that a helper T cell has a different type of MHC molecule, which makes it so that it won't try and destroy the macrophage who's just presenting what it is that it's eating at the site of infection.